The first 10 slides of this presentation are on slides that we already took notes on in class. I just thought it was worth replaying just in case you may have missed something on your DNA note guide. Okay, so now that you're familiar with the structure of DNA, we're going to talk about one of the major functions that DNA is involved in. First off, every time new cells are produced, the new cells need their own copy of DNA from the parent cell to make sure that the new cells are exactly the same as the parent cells. So DNA has to copy itself. And in order to do that, it must undergo a special process called DNA replication. Replication is the DNA copying process that allows the new cells to have a complete set of DNA within their nuclei. The process is quite complicated, even though when we summarize the steps it looks like it's pretty easy. It is quite complicated, and so there are many enzyme helpers to help speed up the process. But the one enzyme that I really want you to focus on is called DNA polymerase. This is the special enzyme that joins the nucleotides together to form that DNA molecule. Now, how does replication take place? There's 4 basic steps. The first step, DNA has to untwist from the double helix because in its normal state the ladder is twisted, so it has to untwist to make it easier to work with. Then, step number 2, the DNA unzips. It unzips at the chemical bonds in between the base pairs. So it unzips, the base pairs are simply like the zipper, and you're just going to imagine a zipper going all the way down through the base pairs to separate the 2 strands. So in step 3, you've got these loose nitrogen bases. They don't like to be singled out, and they like to be paired off with their respective partners, A and T and C and G. So we've got new bases coming in, and they pair up according to the rules of base pairing. And so now you are left with step number 4, 2 new identical DNA strands are produced. So here is DNA replication in a picture format. The old strand on the left-hand side, notice how it's all in purple. It has already untwisted for you. Then, the second step, the purple strand unzips at the bases. Step number 3, the green new bases are going to come in and join their base pairs. And in the end, you have 2 identical strands. They are mirror images of each other, but they are still identical. And notice that the new strands that are produced are half of the old strand and half of the new strand. If you have that scenario, you know that your DNA replication was successful.